Welcome to Brain Ponderings, your window into the world of neuroscience research. Today we're going to talk about aging, which applies to the brain, nervous system, as well as any other organ system. And my guest today is someone who's worked on aging, starting in yeast, looking at molecular mechanisms, involved in some discoveries there on basic mechanisms of cellular aging, which seem to apply to all creatures that we know of. And then, but he's also had a lot of experience at all levels of aging research, either directly or indirectly, uh, up to human research, and somewhat on diseases of aging. And we know that aging is the major risk factor for all the major diseases that kill us. And even if we don't have a specific disease, as I know now, I'm getting older. Brian, you're getting older. I don't know if you have any aches or pains or things cropping up.
Not yet, but it's coming.
So Brian Kennedy, he's a distinguished professor of biochemistry and physiology at the National University of Singapore. So we're on 12 hours time difference now, so it's getting late for me, and it's Brian should be bright and energetic right now. It's
Got
morning.
my coffee and
Got
ready
his
to go.
coffee. And he's also there in Singapore, the director of the Center for Healthy Longevity Translational Research Program, and director of the Asian Center for Reproductive Longevity and Equality.
Yeah, I wanted to call that second one all about ovaries, but they wouldn't let me. So
Okay, but that's kind of what it is,
that's
right?
what it is.
Yeah. Well, it's very important. Uh, women know what
Yeah,
we
it's
don't.
amazing.
There's this andropause idea, but not so much, I don't think.
Not as much. And I was shocked when I got asked to be director of this center. It was a gift by this Via Echo Foundation and from the U.S. And I didn't know much about it, ovarian aging or anything. In fact, I went back to my lab and someone said, somebody figure out where the ovaries are in a mouse. So I don't even know. But having looked at all the research, it's amazing how little research is done on ovarian decline leading to menopause. And we look at postmenopausal symptoms, but we don't look at the ovaries that much and how the what gives rise to the menopause. And so we're excited to work on that. I think that's an underrepresented facet of aging research.
Yeah, I agree, 100%. Now, why don't you start by talking about where did you grow up, Brian?
I grew up in Louisville, Kentucky, and
Oh,
I
really?
I lost my accent, but I can still say Louisville. So
Did you go to the Derby any?
a couple times, yeah, 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 not that much, but once in a while.
That was this most recent Saturday. I have an interesting story. I don't want to take much time myself, but I grew up in southern Minnesota on a 100-acre farm, and my father and my grandfather trained and raced harness horses, trotters and pacers.
Yeah, yeah.
And then, so I went into, actually, I was going to be a veterinarian. I didn't get into vet school, so I became a scientist. Actually, in hindsight, I'm kind of glad I didn't get into vet school. But
Some things happen for the best, I guess. Yeah.
but then
I'm sure you did a great bet, though.
after I did my postdoc, I was applying for faculty positions at different places, and one of the places I interviewed was Lexington, Kentucky, at the University of Kentucky. And I took that position at the Sanders Brown Center on Aging there. And the first couple of years I was there, we had a horse training at the track in Lexington. I'd go in on the weekends and train it, and then we paid
That's
someone.
amazing. Wow.
Yeah.
That center doesn't get talked about that much, but there's some really good aging research going on there, I think.
It's and from the standpoint of brain aging and Alzheimer's disease, they were one of the first NIH-funded Alzheimer's research centers, and a lot of interest. The Nun study, which actually that I don't know if it petered out or not, but
I
uh,
don't know what happened to that study either, but it was really informative.
yeah, there's some good people there. And I don't know if you ever came across Bill Marksberry, maybe not because he's a neuropathologist, but he was he was my mentor there. And he was focused on Alzheimer's, a neuropathologist, went all to to all the autopsies and personally took out the brain tissue himself. And he had a lot of connections with the state government legislature, and he got a bill passed that like a significant, I don't know what percentage, a large percentage of money from drunk driving, speeding tickets went to brain injury and spinal cord research.
Mhm.
And but anyway, so
Yeah, interesting. Yeah.
okay, so then you grew up in Louisville and then you went up to Chicago for your undergraduate?
Did what Northwestern for 4 years, yeah, and that's where I got excited about research. I worked in this guy Rick Gaber's lab who was doing yeast molecular biology, and he was very helpful. And also there was a guy Mark Vidal who later has gone on to a very prominent career at Harvard doing research. But the two of them were so excited about science and yeast that it just got me hooked on yeast, and so I stayed in working on yeast for about a decade or so.
Can you talk about the the things you can do in yeast that are hard to do or you can't do very quickly in other things?
I think for me, the most exciting thing about yeast was it was just so creative. It helped you learn how to come up with ideas and experiments and because you could think up something to do and do an experiment in a week and then have a new result that you have to think up a new experiment. And particularly when we went to MIT and applied it to yeast aging, it was a almost a brand new field. There were a couple labs working on it, but not much. And almost everything we did required a new protocol, a new idea, and it was really a good learning experience for me. I think from the perspective of the value of the organism, it's fast. You can do things genetically that are really unimaginable in other systems still. And pretty much anything you want to do in yeast, you can do. They have a pesky cell wall that's hard to deal with once in a while, but other than that, it's a great organism. And I say this because it's you can't almost can't have a yeast lab anymore. It's almost even before this current administration.
Yeah.
The funding for yeast research was declining. A lot. The labs are not solely doing yeast anymore. And yet, we talk about things like systems biology and trying to understand a cell from a holistic perspective, then this is probably the best place to try to do that. And so I really feel like again, prior to January 2025, even before that, funding for basic science has been drying up,
Yeah.
and it's really to the detriment of human knowledge because I love all the translational stuff that's going on in aging and other places, and I'm very supportive of that. But if we don't keep the beginning of the pipeline flowing, those big discoveries in the future aren't going to happen. It looks even worse now. So
Yeah,
it's
and we're going to talk about aging, but so you got all these young, highly energetic kids, I guess you and I would call them kids, young adults,
yeah,
and
yeah,
they some are going in graduate school and science and postdoc, and so they're at their high level of creativity anyway then, and energy actually, physical energy, mental energy, how long you can devote
yeah,
the effort. And then that's where the basic research is so important. What are the basic mechanisms? And you're right, the NIH, it's if it's not disease oriented, if there isn't some relevance to a disease, well, aging's okay. They're interested in aging research. All these
yeah.
all these Congress people are over the
Getting
hill
old.
anyway, so are on yeah, over the hill.
I'm losing faith I can help Mitch McConnell anymore.
Yeah, Kentucky. Well, he's retiring.
He's
Uh,
retiring anyway.
so in the East, uh, as you mentioned, the genetics are powerful. You can uh, get data back quickly, then go to the next experiment, next manipulation. And some of the genes, like autophagy, is a really good example, I think, where all these different genes involved in autophagy were discovered in yeast. They're just huge. You can see them under the light microscope, these giant, uh, autophagic vacuoles or,
Vesicles and lysosome. Yeah, it's
yeah, yeah,
the vacuole, I guess, at least.
yeah,
Yeah, it's amazing. And there's a Nobel Prize for yeast
yeah,
autophagy and also for secretion, and so I think that, and cell cycle, I think that there's these basic processes were almost completely
yeast.
delineated starting in
Yeah,
yeast, and then knowledge from other systems got added on to that. And then, now, like I said, systems biology really he should be leading the way there. But I don't recommend my grad students or postdocs work in yeast anymore, or at least not solely, because I just think it's from a career perspective, it's very difficult.
yeah. Okay, so then at MIT you were with Len Guarante, and
Yeah,
you're working on protein deacetylases, or is that what you're working?
he was. So he was working on transcription, but he was also sort of at that point in his career where he got tenure recently and wanted to go in a new direction. And there was another graduate student, Nick Austriaco, who since has become a priest. I just visited him in the Philippines. He's apparently one of three priest biologists in the world, so he's an interesting guy. And um, the two of us, Lenny said, "Yeah, do something new, come up with some ideas," and we came up with programmed cell death in yeast and aging in yeast. Actually, we thought aging was less infeasible than programmed cell death. I mean, since yeast is a single cell organism. But in fact, there's a whole field on programmed cell death in yeast now too. So I guess either way we would have gone would have been okay. But we started on aging and through hard work and opportunism, a few good ideas and some luck, we got to the sirtuins. And so that really started that field of aging and Sirtu and the Sirtuins. I don't know, I still don't know that we understand the end point of all that research, but it's certainly become one of the major veins of aging research.
Yeah, yeah. We worked before I retired from the NIH. We briefly worked a little bit on mitochondrial SIRT2 and SIRT3, which seems to be very important in probably most any cell, but in neurons, we found some very
Yeah.
interesting things with it. Um,
I think everybody got focused on SIRT1 because it looked the most similar to SIRT2 by sequence. But I think SIRT3 and SIRT6, there's better evidence for regulating aging than SIRT1, in my opinion. So,
yeah. Now I never worked with yeast, but they kind of fascinate me for a lot of reasons. One is that so they have these asymmetric divisions, right? And
yeah,
this is a common theme, it seems like, where um, even if you look at yeah, I think like egg uh, egg cells. But anyway, you get asymmetric division, and they kind of sort pristine, undamaged molecules to one part before they divide, and then the damaged components to another, and
yeah,
then so one of the daughter cells is very healthy and goes on to divide, and the other one may die. And
yeah,
that seems to happen with mitochondria too, right? They segregate bad parts to one end of the mitochondria, and the good stuff stays on one end. Then they split, and the bad stuff is sent to the lysosomes, the autophagy.
yeah. The common theme in biology, I mean, so many even adult stem cells, when they undergo division, they produce one cell that maintains, potency, and another cell that different starts on a differentiation path, and they segregate proteins in different ways. So, I think that there's been a lot of work in yeast on that with respect to aging and just generally how do things get asymmetrically segregated. So like the spindle pole body always stays in the mother cell, the old one. Damaged proteins are often kept in the mother cell, and there's a range of processes that control this. Also, these extra chromosomal rDNA circles that we studied that accumulate only in mother cells. They're associated with nuclear structures that keep them in the mother cell when the cell divides. So there's been a lot of work. Some of it is just um, diffusion, trying to get through a narrow bud neck, and so there's a lot of new synthesis going on in the daughter, and a lot of old things stay in the mother. But there's also active processes that segregate things in different directions, and we still don't know enough about that, and especially how that works in a mammalian cells where it's much harder to distinguish.
And then you stayed in Boston for a postdoc right after you got your PhD,
Yeah, I
and that
went to
was more cancer related or
yeah, I
not?
went to, and well, it turned out to be aging related in the end. I
Uh,
went to MGH Cancer Center, and I wanted to study with Ed Harlow. Yeah, Ed had this policy at the time where you don't pick up a pipette for a month. You just think about what you want to do and then you do it. And so I was really curious about the retinoblastoma protein, but from a different perspective, which is where is it localized in the nucleus, and how is that relevant to its function? And we did a lot of work that led to studies looking at where replication happens in the nucleus and how that's related to E2F and RB. But it led to one study where we realized that nuclear lamins were controlling distribution of RB in the nucleus as well as replication. And at the time, lamins were just starting to be discovered, especially A-type lamins for having mutations in a wide range of diseases, muscular dystrophy, dilated cardiomyopathy. And later on, they got identified as being a target for mutation in Hutchinson-Gilford progeria syndrome. So, I ended up working on lamins a lot, especially when I was assistant professor. And, I went to it's funny, when I went to the University of Washington, I started working on replication and nuclear organization. And about a year later, I said to my department chair, which is Alan Weiner, who was also a great guy, I said, I want to go start a second project on aging in yeast because I think we missed a lot. There's a lot more to learn from yeast aging. And he said, well, I think as an assistant professor, you should have the freedom. You've earned the freedom to do whatever you want. However, starting a second project this early in your career when you haven't got the other one funded well yet, maybe that's not a good idea. And so I kind of ignored him and went on and did both. To his credit, he didn't get mad about that. But then a year later, the Francis Collins reported that mutation in lamins affecting aging. And so I went back to Alan and I said, look, I was working on aging all the time. I just didn't know it. So
Yeah, yeah, yeah. The premature aging syndromes are where I was at NIA. Will Bohr was there
yeah,
working on DNA repair, and
yeah.
there's certain mutations in DNA repair proteins that cause premature aging syndrome. Why don't you go over, as long as we're kind of bouncing around different things that are going on with aging, maybe just talk about briefly the so-called hallmarks of aging that seem to be occurring throughout tissues and organisms.
Yeah. So, yeah, I think that, one of the things that really moved the cancer field forward was this paper that came out around 2000 by Hanahan and Weinberg, I think, that was on hallmarks of cancer. And it really sort of galvanized the thinking of the field as to certain events or molecular changes that have to happen that lead to a tumor and what leads to metastasis, etc. And so around 2013, Carlos Lopez Oten and colleagues wrote this Hallmarks of Aging paper. And at the same time, the NIA was organizing a conference on on 7 different topics in aging, trying to bring people together. And then they had a day discussion after that conference, and they titled these 7 different things the pillars of aging. And so I got asked to write a review on that. And they told me to synthesize. There were 14 professors at that roundtable meeting, private meeting, and they said, "We want you to synthesize everyone's opinion into a 3,000 word review." And it's like, I do not recommend this. So Claudia Franceschi wrote me like 2,400 words on inflammaging alone and said all of them are essential. So but anyway, we published that in 2014. And so those 2 papers were, I think, had the same effect on the aging field as the hallmarks had on the cancer field. And they really allowed people to start thinking about aging from the perspective of different pathways that were there was good evidence for. I also think they've been misleading a little bit, and that's kind of been corrected over time. But one of the things that's pretty obvious when you look at aging is that all of these things are entrained to each other. So if we talk about senescence, and we talk about telomeres, and we talk about adult stem cell function and proteostasis as being different facets of aging. But when one of them goes bad, they all start going bad.
Yeah.
And when you have an intervention that slows aging, it typically doesn't affect only one of those processes. It affects all of them, at least indirectly. And so I think that I've started thinking about aging as sort of or health, I guess, is a homeostatic network that's there to adapt to things that happen to your body and keep you healthy over time. And when this network breaks down, it gets seen as disruptions in metabolism and
Yeah.
disruptions in adult stem cell function. But it's really the connectivity in the network that keeps you healthy. And also, when the network breaks down, that's when you start getting various diseases. So I think most of the interventions that are slow aging are really preserving that network function, like rapamycin and some of the other ones. So, um, and when you treat an animal with rapamycin, they don't suddenly not have DNA damage, but all the other hallmarks still happen. They have a a modest delay in all of them, and so
Yeah.
I think that's telling you something.
I agree 100% with that. There's not going to be any silver bullet. I mean, rapamycin, it extends lifespan some in mice. It's not dramatic. I don't think it's going to it's not something I would take right now. I mean, when I look at that, there are a few things that we can be confident will extend healthy aging, healthy lifespan. And these are things that they affect multiple pathways, and the reason they do during evolution, these various pathways evolved to respond adaptively to things like food scarcity or the need to exert physical effort
Yeah,
and so on. And so during evolution, individuals whose bodies and brains function very well in like a food deprived state when it had to perform well had an advantage. And we can talk about this later maybe, but I'll mention it now. Seems like more and more there's evidence that what seems to optimize health is kind of challenge recovery cycles, a metabolic challenge like exercise or fasting, and then a recovery period. So during the challenge period, you get activation of adaptive stress pathways, conserve resources, recycle. The mTOR goes down, so the cells they're not in growth mode during but then during the recovery period, the cells go into
yeah.
growth and plasticity mode. But if you don't cycle between those two, that's not good. So you have to have both the challenge and the recovery.
I think that's true, and I think it's also very informative that something like fasting is a challenge that leads to that response and is good. But you could ask the question, why is binging on McDonald's not a challenge that leads to a good recovery? And I think that that comes back to evolution, right? We have a lot of processes that have evolved to deal with low energy states because we had a lot of time in low energy
Yeah.
states. And now we're extremely maladapted to our environment. And when we get into an excessive energy state, we don't have those response pathways to trigger,
Yeah.
a normal, metabolic response that we do for fasting. And so I think it's very informative that that's why we have such an obesity epidemic. We're just very maladapted to our current environment.
Yeah, these pathways get downregulated or not upregulated when you're, in a constant positive energy state from overeating or and you're not exercising. So then the cells kind of, I use the word, they become complacent
Yeah,
and therefore more vulnerable to their stresses associated with aging.
yeah. And in a hunter-gatherer state, when you did have excess, maybe it was good to have that response. You
Yeah,
certainly want to get calories, and you probably want to store calories somewhere for future problems.
yeah.
But now it's too much, right? So, and
Although,
your body doesn't respond to it.
of course, there are only a few hunter-gatherer groups, I think, left in the world. But my understanding, and they're all, I mean, if you look at them, there's none of them overweight. They're
No.
all fit. So I think, they're maybe it's just they don't get enough food, or there's something else about the, yeah, I mean, they're not eating at McDonald's, that's for sure. And
Yeah.
they have to move physically to get food.
Yeah. For me, I do a lot of exercise. I always have, but I would give myself maybe a 6 out of 10 on diet. Not great.
Hmm.
And so I made a few changes about 6 months ago, and maybe went to 8 out of 10. I'm not sure I can get to 10 out of 10, but I got maybe to eight. And it's amazing the effect that changing your nutrition can have. I lost 10 pounds, more energy, I'm exercising better, and so it just reinforced to me the sustainable, even modest changes in how you affect your diet, but as well as exercise and sleep and stress or have such big impacts.
Yeah, and it can improve your productivity, right? So I eat like a blue zone diet, mostly complex carbohydrates from vegetables and some fruits, not a lot, but some nuts, meat, mainly fish. I don't eat a lot of red meat. And then what else? Whole grains. I think whole grains are fine.
Yeah.
Um, yeah. And
One,
what I do is I don't eat breakfast. And then
yeah,
I exercise at the end of the morning before I eat lunch. And I find
yeah.
my brain works the best in the morning when I'm in a fasted state.
Yeah, I think I don't always eat breakfast either. And when not traveling, I try to eat, one big meal a day. I'm not completely compliant to that, but I try to do that. And I think for me, I've also recently started eating a little bit more protein because I've been doing a lot more resistance training. And I think that's one thing that we don't. I'm not a keto diet person by any stretch of the imagination. I would tend to eat more than the direction you do. But I do think we don't pay enough attention to the interaction between these behaviors, like
Yeah.
exercise and what we eat. And we get into these big arguments about what's healthy from a nutritional perspective, and we don't consider the fact of whether we're talking about a sedentary person or an active person or
Yeah.
a lifting person. And of course, a lot of people lifting weights are pouring tons of amino acids down, and that's almost certainly bad.
Yeah.
I'm not suggesting huge diet of amino acids, but I think modest changes might be trying to optimize a balance when you're doing those things is probably helpful.
A while ago, maybe a couple months ago or so, I did a podcast with Dudley Lamming,
Yeah, Dudley, he's
and
a good friend.
yeah, and I had a lot of number of comments of viewers, kind of essentially saying, "What are you talking about? You need to get a lot of protein," and
Yeah, yeah,
but he was just saying what the animal data show, and So we get back to this idea. It's not good to have the mTOR pathway constantly activated. That's not a good thing. And
Which is what's happening in aging, it looks like, at least in some tissues. So,
yeah, but muscle mass maintenance, but you have to have sufficient protein, obviously, to maintain muscle mass. But exercise is the key thing. Your muscles, no matter how much protein you eat, if you don't exercise, your muscles aren't going to get
yeah.
bigger and maintain their bulk.
Yeah, I agree with that. And I used to be a runner only, and I switched to doing running and resistance training, and I've had huge benefit from that, I think.
I wish I would have. I should have done resist. So when I grew up I started running in the 70s, right? And the elite runners there, um, all they do is run, right?
Yeah.
Now they do some cross training and stuff, but the training has changed over the years.
Yeah, it has. And I just know for me that it helps my running when I do resistance training. Yeah, they benefit each other, but also I just feel better, and
Yeah.
having extra strength and some upper body strength is just I think it helps a lot of facets of my life.
I've got the
There you go. We should do that while we're talking. We could do the next podcast from the gym.
No, I'm pretty weak. I had a lot of issues. I had a mountain bike accident, a number of surgeries, and then it's taken me a long time to get back. And then I found out I have a mutation in a voltage-dependent sodium channel that's expressed in nociceptive neurons,
Uh-huh.
and it's a gain-of-function mutation. So I have this pain syndrome. It's called primary erythromelalgia, where I have hypersensitivity to pain. And so combined with those injuries, it's been
Makes
kind
it
of a
difficult.
long process getting, but I'm back now. I'm back. I do 30 minutes pretty hard on the stationary bike, and I walk for, I don't know, half hour straight plus some more every day. And then I do some resistance training, but not with really heavy stuff because my injuries were in the core,
Oh, yeah.
the adductor muscles and where they attach to the pubic bone, and I got to be careful. Uh,
I had
Everything you
some.
do is connected to the core. You can't hardly make any move without affecting.
I had sort of the opposite. I had a birth defect that was something close to Hirschsprung syndrome, where part of my colon didn't develop, and
Oh,
uh, no opening at the bottom, and and so they had about 7 surgeries over time to fix that. And then when I was 22, I got in a car accident that fortunately I didn't injure my spine, but I injured everything else. I tore my nose off and broke 14 bones and spent 10 days on a ventilator. And I've got a lot of metal inside me, but it's a year later. After it took 6 months before I could walk, but within a year I was sort of pretty much have full health from it. I don't have any long-term effects. So so far I've had all my challenges early, but I'm sure there are more to come later. So
well, I think we tend to get more careful as we age. Hopefully.
I'm kind of getting less careful at the moment. So
Okay.
I've become my own experimental organism these days, trying different,
Well, it's interesting.
supplements and drugs.
Although all the organ systems they age, but the brain, as you get older, you think you're young still, and you don't realize your reaction times are getting slower, and you're not quite as strong or as quick or as,
Yeah.
and yeah. All
Nobody
right.
thinks they're old. I think. My grandmother at 97 was like, I don't want to buy that new color TV because I may have to spend 15 years in a nursing home. She was still living at home in her house at the time. So I'm like,
Well, that's a good
what do you
positive
say?
attitude.
I'm like, buy the TV.
Okay, all right, so these hallmarks of aging, oxidative stress, DNA damage, impaired autophagy, um, inflammation, et cetera, et cetera. Yeah, mitochondrial dysfunction is a big one, perturbed energy metabolism, and so
Yeah,
on. And they interact in experimental systems, if you impair mitochondrial function, that's going to cause increased oxidative stress and a lot of other things, et cetera, et cetera. Um, can you talk about, so up in Seattle, right? So you were involved in the dog study, right?
Well, only
Uh,
loosely. So I'm on the advisory board that helps out with
Okay.
the dog aging project. You know, Matt Caberline and I met in Seattle when I went there as an assistant professor, and he was
Ah,
a postdoc.
ah,
And so, you know, we started collaborating first on yeast and then on worms and mice and a bunch of other things, and most recently dogs. And so he got very interested in the dog aging project. He's still involved in that,
yeah.
although the rapamycin trial is being done, I think, by Roz Anderson now. And I think dogs are fascinating for aging research, and I'm talking about companion animals, not so much dogs in the lab. And Because you know they have so many properties that mice don't have that are related to how we age. They live in your house, they sleep in your bed, they eat your food. They probably shouldn't be eating your food, but they eat your food. You probably shouldn't be eating your food either, but that's we've discussed that already. The But anyway, they I think a lot of what we learn about dog aging is going to correspond better to human aging than some of the lab animals that we spend time working on. Those are still valuable. I'm not trying to say we shouldn't work on mice, but I think there's a unique opportunity to learn from dogs.
Yeah, I agree with that. And from the standpoint of the brain, they're very interesting too,
Yeah.
right? And there hasn't been enough research on anybody that's owned a dog knows that they're very smart. They learn things pretty
Yeah.
darn well and remember them pretty darn well. They have emotions. We always had, I mentioned I grew up on a farm in Minnesota. We always had two dogs, mostly German Shepherds. And a couple times when one of the dogs would die, one case was cancer and another case got hit by a car. Um, and then when one dog died, the other one was clearly depressed.
Yeah.
I mean, it's so obvious,
Yeah.
right? And yeah, so they have a lot of human-like traits. I often wonder, we shouldn't anthropomorphize things, but to me, and this is particularly true in behavioral neuroscience, that things like emotions, like empathy, joy, whatever, pleasure, etc. Um, the people who work on that tend to say, well, that hasn't been, we don't know that's occurring for sure in these lower animals. But I think that, we're just one species. Yes, we're very
Yeah.
smart and so on, but shouldn't kind of the default be to say, assume they do have that and then try to prove they don't?
Yeah, it's true. I mean, look, I think you're right. Anybody's had dogs long term, and I grew up with dogs. And you know, when my kids were growing up, we had a Newfoundland and a dog that was like a Great Pyrenees that was a Slovak Chuvach. It's a Hungarian version of a Great Pyrenees, a male and a female, and they were very close to each other and my kids, you know, and so that
Uh,
and you could definitely see that, especially the female, the white dog, she had a lot of emotions and a lot of attitude, and she was funny because she was super friendly with other dogs, but the minute we had kids, they became the sheep. She was a sheep guardian. And when another dog got anywhere near those kids, she was like, she never
yeah,
hurt another dog, but she definitely was aggressive and was like, you're not getting near these kids. And so we had to be very careful with her after we had kids. It was funny. The Newfoundland was just a happy dog. So,
yeah. Uh, Go ahead and talk about your experience in Singapore. First, talk about Singapore. I actually got invited there a couple of times, but I didn't make it. I had former postdoc. Did you ever come across Gary Aramugam?
yeah, yeah, we collaborated. So we had some papers together. He's
Yeah,
down in Australia, but it's
yeah,
he's a really good scientist. Yeah. I think
yeah.
we may be co-authors on one or two of those papers. I have to go back and
It
check.
could be. Yeah. Right, but so Singapore is very, in some ways, unique.
Yeah, it really is. It's you know, historically, this is a country that got its independence around 1965. Don't quote me, but it's within a year or two of that. And got a leader, Lee Kuan Yew, who set up a democracy, but his party has won every election since then. He was the prime minister for 50 years or so. And he really set about establishing a country that cares about the population and tries to build a great environment for people to live in. And that doesn't mean I agree with everything that the government does, but I think one thing I will say that is that I do believe this government cares about its citizens, and that's more than I can say about my own government right now. So, people have economic, um, comfort here. They're all highly or mostly highly educated,
Yeah.
and most people trust the government, and so it's a very compliant population. And you can see that in the COVID epidemic because that the government said they had pretty serious rules about not we couldn't even visit somebody in their other house, and there was a really strong lockdown for a period. And they said once we get to vaccination, that's 90%, we're going to really relax the rules. And of course, they got there quickly because the population believes the government, And if you said you could say that, don't jump off a bridge, and half of Americans would jump off a bridge. And it's really um, it's unfortunate. But here there's an opportunity to really establish a healthcare system that is very effective and geared toward healthy aging. So the idea of doing healthcare and not only sick care is really emerging here. It's growing. The government's getting behind it. They have a huge need to because the life expectancy is extremely long, maybe the longest. They're certainly in the top three, and the birth rate is extremely low. It's 0.9. So there's no placement basically. So basically, the huge percentage of the population is going to be over 65. It's becoming like Japan rapidly, and there's not a lot of room here to bring new immigrants in either. It's a small island.
Yeah.
So
Hmm.
you got to keep people healthy. And I think that there's a recognition of that and a real movement toward trying to accomplish that here in Singapore. And so I'm happy to be here trying to, provide the science behind some of what needs to be done and also to translate it. So we have a large program now where we're doing human clinical studies. We're looking at community-based studies, also trying to promote the longevity clinics and the industry a bit. And so it's exciting times for the aging field in general, but particularly because we think Singapore could be a, sort of a benchmark for what could be accomplished if you really focus on keeping people healthy.
So, and yeah, it's very different than here where the healthcare systems, it's all for profit and there's virtually no attempt to for prevention, risk reduction. You go to a primary care doctor, whatever, they take your blood pressure, do your basic blood work, and if you're
Yeah,
overweight or your blood glucose is getting up, they'll just tell you that and tell you oh, you should exercise, don't eat so much, and then come back next year. And of course, nothing's going to happen. And then in the meantime, people are getting bombarded with ads, fast food ads, ads for drugs all
yeah,
over the place in the United States. Prescription drugs are with all these happy people with, some severe condition that they shouldn't be happy, but apparently the drug's making their symptoms
yeah,
going away, which isn't true, and
yeah.
yeah,
It's shocking, when you come back because they don't allow those type of commercials here in Singapore. So, and then when you come back to the US, you just don't understand how bombarded people are with those kinds of ads. I'm not I work with pharmaceutical companies. I think these companies are filled with people that are trying to do good for the vast majority of people in those companies, and certainly they've developed effective drugs, but the over reliance on these drugs when lifestyle could make such a big difference is a concern, I think. The US is just a perverse incentive system. It's like I had a CEO of a hospital chain who visited me when I was CEO of the Buck Institute, and he was looking to give money to something. And so we talked about health span and healthy aging. And he literally looked at me and said, "Why would I give money to you? If you're successful, you're going to cut my procedure rate at the hospital by 60%." And I didn't really have an answer to that, but that's, how
Yeah,
and they're not like that. I have to say that wasn't Kaiser. I think Kaiser's a little bit more forward thinking than a lot of them, but the when you're driven by profit ahead of health, there's a problem, and I think the U.S. system is still dominated by that.
yeah, in an ideal world, the healthcare, anybody, whether it's a physician or whoever in healthcare system, their goal should be to put themselves out of business,
Yeah,
right?
I agree. And you look at here, they've got a sort of public-private system where there's a public healthcare system for everybody. If you want to pay more on
Uh,
top of that and
uh,
have your own private room or faster procedures or whatever, you can go to the private hospital and pay for that. But even that is affordable. You're not going to like spend $200,000 for 5 days in a hospital, So it's a pretty effective system. It gets around some of the criticisms of the public system alone, but it still has a very strong public system that supports everybody. And probably that's the most realistic way to make something work. And you can see that in the cost is like 7% of GDP for health here rather than 20% in the US, and it's way more effective for the vast majority of people. So,
yeah. What about, all right, so advertising can be a problem, right? Whether it's food or drugs. So what about the internet? I never got on social media when it came out,
yeah.
mainly because I looked at it and say, I don't have time for this.
Yeah.
I got too much to do. I'm not going to spend my time kibitzing with a bunch
Yeah.
of people gossiping. But now there's so much information out there in pretty much any realm of life you can think about. All these influencers who, in many cases, either they don't have a strong background in what they're talking about, they're not experts, or they may be an expert in some area, but then you don't know who they're getting money from, and they're making money from. Uh, See, nobody, hardly anybody watches my podcast because I don't advertise it much or anything, but um, so this is really a problem that whether it's ketogenic diets or protein or uh, you know dietary supplement XYZ, and I don't know. Personally, the only supplement I do take vitamin D and a multivitamin. That's
Uh,
it. Um, yeah, that's it.
yeah, I have to say I work with supplement companies, so I'm not fully
Ah, well,
uh,
if you're deficient in something, then you should take it.
yeah, so that's the first thing I would say is that I think we have not done justice to vitamins because most of the meta-analysis of these vitamin studies have indicated they don't do much. But I think that's a reflection of bad studies more than it is the vitamins. I think
Hmm.
that uh, most of those studies were not done in a way to try to optimize the levels of a person's vitamins. And I think that if you set an optimum range, and we could argue about what that is, but we set some range where we try to look at an individual and get them in that range for A, B, C, D, and E.
Hmm.
That doesn't mean necessarily just taking a multivitamin. It means analyzing every 3 months and changing levels of supplementation, which is not what supplement companies want to do. But if, in principle, we did that, I suspect we would have a big impact on aging in the vast majority of people. I think if you just give everybody something, there's a range they're in, and then it changes the
Uh,
range they're in. It doesn't necessarily make more people in the optimal range. And I kind of feel like these meta-analyses are kind of garbage in, garbage out, and it's unfortunate because I do think these vitamins are important. We've seen big effects I've never published this actually, but we've seen that if you get the vitamin A supplementation right in mice, it extends lifespan in males significantly.
Vitamin
There's,
A?
yeah,
Ah,
there's good data. There's good data on D. I think B complex vitamins are becoming more and more relevant to aging pathways. They're certainly involved in NAD metabolism and, and methylation, a lot of other things. And so
and also,
you
yeah,
know,
the folic acid, homocysteine, one carbon
yeah,
metabolism pathway.
yeah. Every time find a new drug or gene that influences aging, it's somehow related to one carbon metabolism. I can never put the puzzle together to come up with some elegant explanation, but it's always touching on that pathway. So something really important is happening there.
Now, I had some interest in this, and I still do. Why is it that vegetables and fruits seem to be good for health? And one idea is it's not that they have chemicals that are sopping up directly free radicals, but they have chemicals that are inducing adaptive stress responses in cells. In other words, the reason these chemicals are in the plants is to keep insects and us from eating too much of the vital parts of the plants. But we
Yeah.
co-evolved over long periods of time
Yeah.
so that we have multiple mechanisms. That, Another interesting thing about these naturally occurring phytochemicals is, as you know, there's enzymes in the liver that rapidly degrade them. So they're not in our system very long compared to some man-made chemical which we didn't evolve with. So you only get
Yeah.
transient exposure and they induce mild stress response.
Yeah. No, I think it's really interesting. The One of the things I changed in my diet was to try to eat really diverse, and that's easy here in Singapore because the food is really diverse anyway. You can eat, high quality Japanese food one night, and Korean food, and you can get all kinds of vegetables and a whole range of fruits that you don't even have access to easily in the rest of the world. So it's a good place. I realize that it's expensive to do that, and that's not a solution for everybody, but that's something I've done that's I think really helps my health because I get exposed to a lot of different kinds of phytochemicals. Obviously, I think that fruits are better. You would agree, I'm sure. Fruits are a much better way of getting your carbohydrates than sugar directly.
Yeah.
And because it takes more time to break down the sugars in most fruits. Although it's funny, I was in Saudi Arabia recently, and I was traveling with someone, and they have one of those CGM monitors on,
Yeah.
and we started eating the dates in Saudi Arabia,
Oh,
and
date.
I'm like,
Yeah.
wow.
Yeah.
You want a glucose spike. That not all fruits are necessarily good for that, but some of them are. And so it's yeah, I think that really helps. And speaking of that, I think everyone should wear a CGM, not all the time, but just periodically, because it's not so much that it's valuable for your doctor, it's valuable for you. You learn from doing it. You spend 2 weeks eating different foods and see how you respond. And people respond differently to different types of foods. And understanding what drives your glucose up and what doesn't is probably useful for how you arrange your diet. So I think it's an educational tool as much as anything. And
That's a good idea. I haven't done that. I may try that. I know you mentioned dates. It's really interesting. There's these bars called RX bars that
yeah,
they have dates, almonds, cashews. I get the blueberry ones, but the
yeah,
dates, when I eat something that has, high amount of simple sugar, relatively simple sugars, I can actually feel like I get hot.
Yeah,
I don't
yeah,
know
yeah.
what's going on there, but
You
whereas if
notice it.
yeah,
You get that with the dates then?
yeah, with the dates.
Yeah, yeah. Those bars are, I've started eating a little bit of them because, as I said, I'm trying to get a little bit more protein without fat, and so um,
Oh, it has egg whites too.
yeah, you look at these things though, and some of these, I won't name names, but some of these bars don't look healthy. I mean, they're adding sugar to some of these bars, and you really have to dig through and find the ones that have the ingredients you want because they're all in this health section of protein bars, but the composition is way different from one to the next.
Yeah, yeah. What are your thoughts about ketones and particularly BHB beta hydroxybutyrate? It's pretty
Yeah,
clear now that this ketone acts as a hormone. It's activating signaling pathways and it's not just an energy substrate for cells.
yeah. I think I bet by the end of the day we'll say that about every metabolite. but but yes, I think this one's important. To me, ketogenesis is it's important how you get there. And if you're getting there by fasting or diet or exercise or something like that, or some combination, that's probably extremely healthy for you. If you're getting there by dramatically disrupting your nutrient balance and eating only protein and fats and no
Yeah,
carbohydrates, I think this is not a healthy way to do it. I mean, you probably will lose weight, but it's hard to sustain it. And in long term, I just don't think it's healthy for aging. So,
yeah,
um, I'm supportive of the idea of increasing your ketones, but I think it's important how you achieve it too. So,
yeah, I agree with that. Okay, let's see. So in Singapore, are you do you have input to the government? I mean, what kind of a new initiatives do you think would be good even in Singapore, even as they already have a lot of good things going on?
yeah, I think I talked to people in several different governments now, and the good news is that different governments around the world are starting to really think about healthy longevity, and that hadn't happened for a long time. I think there's some thought in the UK, but also places that you wouldn't naturally think to lead the way. There's a lot going on in the Middle East right now, in Abu Dhabi, in Dubai, Oman, Saudi, where people are thinking about the health of the population and how longevity science can apply to that. I think that's really exciting. And then Singapore, Singapore was really early to identify aging as a problem. I got here 8 years ago, they were already talking about it, but they've taken a lot of time trying to figure out how to deal with the issues. And, a lot of the early stuff was really helping manage older people rather than keep people biologically younger, it was what do we do with our Alzheimer's population? And maybe we should put better signage up so they don't get lost and stuff like that. None of that's bad. I don't have a problem with it, but it's not really solving the aging problem. And I was out, when I do interviews in Singapore, I'm always saying, because also Singapore has this, the Chinese population in general has this idea that when your parents get old, the younger generation takes control and takes care of their health, which is the opposite of what happens in the US, I think. And sometimes the younger generation gets overprotective of the older people, and so I'm always like on interviews saying, let your parents go out and play. It's like
Yeah,
don't keep them in the apartment, and especially in Singapore, there's no crime. If they get lost, somebody will bring them home. It's not a big deal, and I think that the that's a bit of a challenge here. But now they're really focusing on moving the interventions earlier to middle age, people still working, and trying to eventually, I think, restructure the whole society around what it means to get to 65 in terms of your not just your biological health, but in terms of your life structure. Are you going to retrain for new jobs? Are you going to you know, stay in your existing job. What are we going to do? And they haven't solved everything. I think still we need to. We talk about keeping older people active. We don't talk about keeping them empowered. I think that's really important. They still have a forced retirement here at a certain age. They're relaxing it, but it still exists. And I think that that's the last thing you want to do, is send older people home to sit on the couch, you know. So it's there's a lot of ageism everywhere in the world, not just here. And so I think these things are still need to be dealt with, but they're really thinking broadly about what to do with an older population here. And I think that it's going to yield positive outcomes, although not all the solutions are in place yet.
Yeah, as people get old, in their 70s, 80s, as you said, it's good to keep their brains and bodies active. And kind of for me, like an ideal thing now is I spend half the day doing this, writing,
Yeah.
um, thinking about science, that kind of thing. And then the rest of the day, I kind of goof off, but not really. I mean, I've got a garden. I got it going now. Everything's growing. Do stuff outside. A granddaughter, we got a 2-year-old granddaughter who lives close. She
Great.
comes over 3 days a week. And so that I find this kind of ideal for me at this stage of life. It's kind of
Yeah.
a hybrid thing.
Yeah, that's great.
Yeah.
And I think finding your own path is important. And one of my pet peeves, by the way, is that we need what we call older people keeps changing, and it drives me crazy. You know, it's like, why don't we just call them old? It's like it went
Yeah.
from old to older to elderly to
Yeah.
elders. You
Yeah.
know, pretty soon it's going to be orbitationally experienced. You know, it's like it's ridiculous. You know, it's like the problem is not the term; it's that we associate bad things with the
Yeah.
term.
Yeah.
So what we need to do is to go back to old and make old a positive thing, or at least a neutral thing, and not a bad thing. I think people like to embrace the idea of old. You know, you hear this all the time. I'm old. I'm doing okay. You know, I think we need to support that rather than keep changing the word all the time. It drives me crazy.
Yeah. Well, this is a good place. If you can take a few minutes and talk about something one of your recent articles and with other people you're working with, collaborating with on aging clocks, metabolic
Yeah.
aging clocks. Can you talk about
Yeah.
that a little bit?
Yeah, we've really got into this aging clocks. I think that in 2010, you used to hear people going around saying that, well, we set up the NIE did this huge thing on biomarkers of aging, and it really didn't work very well, and then what happened is you had people start to use machine learning and AI, um, and which is basically at that time, just linear algebra with scale. But anyway, they call it AI. Now it's gotten more elaborate, of course. The idea was to try to model using complex data sets the age of the individual. And so a lot of this was done with DNA methylation patterns. Let me just say primarily that I don't think there's any primacy to DNA methylation. I think that you can do this with any omics data set. You can do it with microbiome. You know, we work with someone in China that estimates biologic age from changes in facial structure from three-dimensional reconstruction. I don't think that DNA methylation is driving aging just because you can measure biologic aging or aspects of it from patterns of methylation. But anyway, a lot of people did work with methylation. The first clocks were really designed to predict your chronologic age, and then the residual error from that was your biologic age. I think those were good as a foundational starting point, but problematic for use in the wild. It's we don't really um, we focus more on the second generation clocks like Grim age and Pheno age, which are designed to predict outcomes like mortality or clinical parameters or something, morbidity. And recently we've been making our own clocks for a couple reasons. One is that mechanistically, methylation is hard because you find 50 methylation sites that change. It's really hard to understand mechanistically what that means for aging.
Yeah.
Whereas if you make a proteomics clock or a metabolomics clock or a lipidomics clock, these are molecules that have actions in the cell. So if you can identify the 12 proteins that predict 90% of the biologic age of an individual, then you can look at what those 12 proteins are doing. You've got a shot at understanding what's going on relative to aging. So we're doing a lot of that. But we've also gone in the other direction and produced clinical chemistry clocks. And there's a couple reasons we've done this. One is we want to get clinician buy-in. We want to get people that are not thinking about aging to start thinking about aging. And the second reason is that the commercial DNA methylation clocks are not very accurate right now. There's if you, and I think they're trying their best. I know a lot of these companies, and I don't want to be overly critical, but DNA methylation is not a standard operating procedure right now.
Yeah.
That how you do it still requires a lot of expertise. You're getting, especially when people are sending in blood or saliva samples that they collect differently, that they mail differently, that they store differently. There's a lot of variability there. So we went back to the NHANES data set, which I'm sure you're familiar with,
Yeah.
and we collected all the clinical chemistry data and questionnaire data, and then we just tried to predict mortality in that population. And it turns out we can, with AI, generate a predictor of mortality that's more accurate than any of the methylation clocks. And it uses parameters that have high consistency, like HbA1c. You measure that in Singapore, you can measure that in the US. The consistency is much closer. And it also has the added value of when you go to a doctor and say, this person is biologically, we're actually predicting mortality with the clock, but you don't tell the client that they're mortality risk. So we use demography to convert back to a biologic age. So when we go to the doctor and say, all right, this person is biologically 5 years older, and it's because these four parameters, none of which are out of the reference range on their own,
Uh,
but they constitute a principal component that together the four things are driving mortality risk, and they're all actionable. That's the great thing about the parameters in NHANES. They're actionable parameters. They're with drugs or with lifestyle, but one way or the other, they're actionable. And so then you can see that you can bring people's risk back down, which reduces their biologic age. Now, I don't think that means we're reversing aging per se, but I think what it's saying is that given the age the individual's at, we're putting them into a healthier state that's lowering their mortality rate. So it's very actionable, and we now have hospitals coming to us saying we want to institute this clock, which is not happening with DNA methylation still. So I think that's exciting for us.
are all of the how many different, six different things that, how many did you say are in
Oh,
your aging
no,
clock?
actually, NHANES has about 160 features, but we narrowed that down to about 50 features. But it's less than that really because a complete blood count gives you 30 different features. So, it's maybe 20 things we measure, all of which you've heard of before, and
Okay.
we've
Right.
removed some of the expensive things too because there's enough redundancy in the data. We can take out things that are hard to institute.
Yeah.
So if you did it from scratch, it might cost $200-300 to collect this information. But a lot of people already get it. They
Ah,
hospitals already measure these things. There are huge data sets and computers of people that have measured most of these parameters. So we can look back at retrospective data in hospitals too. And of course, there's differences by ethnicity and location, and and in Haynes is a historical data set. So there's some adaptation we have to do, but I think from a clinically actionable standpoint, this is an interesting way to go.
yeah. I'll have to check my lab work and see if.
Yeah, I'll send you the parameters. We can do
Okay.
another show where we reveal your biologic age.
Well, maybe depends.
Well,
You
it's a
mentioned
funny thing when you go to a client and say, you're biologically younger, they all say, I knew that. I'm glad that happened. That confirmed what I already knew. When you go to them and say you're biologically older, they say your clock is not accurate. It hasn't been validated.
You mentioned the early biomarker work that NIH put money into, and I remember when I moved to Baltimore and Don Ingram, I was talking to Don
Yeah,
Ingram, he was involved in that. And the one thing they did find, and they published a very short paper in Science, body temperature. So
yeah, it's interesting.
lower body temperature predicted longer uh, lifespan.
Yeah.
Is that held up, or is that?
I think there's some evidence in animals that's true too, isn't there? I
Yeah,
mean,
yeah, yeah,
there's some mouse
yeah.
data and there's some worm data too on that. I think that I don't know in the human, we haven't
See,
done any
I
analysis
want
of that.
that to be an accurate biomarker because I have a my body temperature is always a degree lower than it's like
Yeah,
97, 97 or something like that. Yeah.
yeah. I think it's reasonable that this would be true, right? But I
This
haven't
from a
seen.
physical a chemical reaction standpoint, right? Everything's slowed down a little bit.
Well, we just had the first conference on gerophysics here in Singapore, where we brought theoretical physicists and aging biologists together. And the reason I helped organize that meeting was partly because I got tired of hearing aging biologists discuss what aging is. I'm sure you've been in a million of these discussions where you try to define aging and
Yeah.
you argue for 2 hours and you come up with shit happens and then you die. It's like it's annoying, right? So we thought there's a lot of data now. There's a lot of human data. There's a lot of data. Let's try to bring some people that know how to model things, theoretical physicists, and they model things based on core principles like entropy. And let's see if they can model aging data. And so we're writing a report of that meeting, but I think that we're hoping to drive a new field there because I'm starting to think that we're not going to define aging in a meaningful way until we do it mathematically. And that has to be done based on physical, chemical principles, not on,
Yeah.
random variables that you throw into equations. So I think that, and there's progress on it. There's some interesting ideas on how to measure resilience and we're hoping to really help keep that field moving because I think it's a new area that could solve some of these understanding problems.
Yeah.
But certainly, you're lowering temperature with lower entropy, right? So,
Yeah. 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 Absolutely. And if you lower it enough down to whatever minus 273, you stop aging. Of course,
yeah, that's true. No one would
you're
die
not
from aging.
functioning. But I when I was in ninth grade, we we had to write a essay on I don't know, it wasn't a science course, but anyway, I was getting Popular Science magazine back then, and they had this article on cryogenics, right?
Yeah.
And so I wrote an article on that. So I think that was my first interest in stopping aging. But and then, you
Well,
did
have
a
you
lot
have you
of
tried cryotherapy yet? That you need to try it. You can. There are lots of clinics doing cryotherapy now, and the one I tried was minus 87-degree room for 3 minutes. And so you go in with very little clothes on, cover your ears, put some mittens on, and you go at minus 87. And it was interesting. I think the argument is it reduces visceral fat, and it may. I think that the challenge is that if it's going to work, you need to do it frequently, like 3 or 4 times a week. And most clinics, people don't want to come do that, but you definitely have interesting body sensations when you take your temperature at that level, to that level. And you definitely feel, I don't know, it's very different than going into like 0 degrees. I think you feel worse if you go into 0 degrees. If you go into, because at zero, your body's signaling all these inflammatory responses and you get pain and all this stuff. At minus 87, your body's kind of like saying, all right, I'm doing nothing. I'm going to conserve all the ATP I have and hope this idiot's smart enough to get out of this temperature, And so
Huh.
your body reaction, I think, is very different. Like you feel a sensation that you should be cold, and you do get a little bit cold, but it's not as strong as you would have expected for 3 minutes at that temperature. It's really interesting how
Huh.
your body reacts. I don't know if it's good for aging per se, but it teaches you a little bit about biochemistry at least.
Well, and heat. Oh, we could talk a long time, but maybe let's finish up about talking about climate change and its impact on aging. Uh, so I did a master's degree down in Texas and at a university where they had one dorm, there was no air conditioning. It was a the musicians, all the music majors lived there. And they had
They
fans
put
and
them
stuff.
there intentionally.
Yeah, they're crazy. But you know, yes, the temperature's gotten higher, but it was very hot in Texas before there was air conditioning, right? And it's very well known that if you're not acclimated to a high temperature, you're much more prone to heat stroke than someone who's acclimated.
Yeah,
And actually, there's a recent case. I don't know if you heard of Michael Mosley. He
yeah.
he was a BBC producer. He did a
Oh, yeah.
documentary
I did hear about that. Yeah,
on
he was on some island somewhere.
yeah, in Greece, a Greek
Yeah,
island, and he did it in 2013. He did a documentary on intermittent on fasting,
yeah,
and Walter Longo, me, and Krista Verity were in it, and it was he did it because we'd helped a group in England do the first controlled clinical trial of intermittent fasting in humans. But, so he'd been doing all these things to, improve his health. But he went from the UK and he went to this Greek island where it was like 100, in the shade. And and it was kind of a mountainous island. And he went on a hike by himself and he died of heat stroke.
yeah.
So
Well,
yeah,
it's interesting. Singapore is probably one of the only like highly developed cities on the equator. We're 50 kilometers from the equator here, and the temperature never changes all year. I mean, they say it changes, but by that they mean 1 degree Celsius. So it's like 32, 33 Celsius, about 90 degrees Fahrenheit in
uh,
the all every day, and the humidity is extremely hot every day. It took me, I think, 2 years to adapt to running in this environment. And even now, I was just gone for like 3 weeks, and I came back and I was in Malaysia and went for a run Sunday, and my heart rate was too high. It was like I was even 3 weeks is enough to get me unadapted to it. So
yeah.
it's certainly true that you have to adapt. This climate's really interesting. I mean, the air conditioners everywhere. We joke that Singapore has two seasons outside and inside. But yeah, it's a challenge here, and I wonder what that sort of climate does to aging and how that might be different in this population. But it's hard to dissociate that from all the other parameters. There's different ethnicities here, there's different diets, and so it's complicated science, but
Are
there's
are
definitely
you
a lot going on.
do you get hurricanes, typhoons? Are you prone to that kind of?
Yeah, Singapore's amazingly protected. There are no tectonic plates under it. There are islands like the Philippines and Indonesia surrounding it that take the brunt of all the hurricanes and stuff and the tsunamis. And so it is in the region where all these things happen. There are volcanoes in Indonesia around us, but none here. It is in the region for all of those things. But Singapore is like this little protected zone. And they've also designed it in a really good way because we do get monsoons occasionally, but the drainage, the way they've engineered the city is you might get flooding for a couple hours, it's gone. It's really impressive how they've organized things, but they're also in a good geographical location. So
Yeah.
it's pretty protected here. You should come. You know, you've got time now, so.
Yeah, I should. We'll see. I'm not traveling much lately though, with all this neuropathy's kind of a problem. But
It takes
yeah,
forever to
if I
get.
come, I'd come with my wife probably and
Yeah,
we'd
sure.
spend some time. Yeah.
Yeah, and if you did that, you should go to it's surrounded by beautiful countries. You know, they're all of them are different and interesting. You know,
Oh
Vietnam,
yeah,
Thailand,
yeah.
Indonesia,
It'd
Malaysia,
probably
Philippines.
take a month or something and
Definitely, because there's worth spending a few days in each of these countries. They're all unique, and I love Vietnam, but I like all of them. So
yeah, and I've got, former lab members in a lot of those places.
yeah, I bet you do. We got a lot of those
Like I
things.
could get, have a free tour guide and
Yeah, there you go.
yeah. Okay, Brian. I appreciate you taking the time.
Anytime, happy to do it.
All right. Bye.
Okay, bye bye.